Well, good morning or good afternoon to everybody. I would like to thank the organizers in the name of all the speakers of this hCOMET symposium for giving us a chance to present new data from our hCOMET group. So this hCOMET group is actually a shortcut from the name Human Biomonitoring Alkaline Comet Assay Group. And we will present the use of the method and its new perspectives. So this symposium will last for 2 hours. It will have 4 speakers, 5 minutes for each speaker, and 20 minutes at the end for the questions. And since I'm the moderator, I should represent myself. And after the end of my talk, I will represent each of the speaker after me. So I'm a molecular biologist. I work at the Institute for Medical Research and Occupational Health in Mutagenesis unit in Croatia. And my main research topics are human biomonitoring, genotoxicology, nanotoxicology, occupational exposure, and mostly Comet assay. I was involved as a working group leader of the COST project, COST Action, hCOMET, and the leader was of the project, Professor Andrew Collins. And this project has been finished, so we will present our results. And I have 91 publication in Web of Science core collection. So the title of my presentation is Alkaline Comet Assay and DNA Damage Assessment in Frozen Whole Blood Samples, frozen without the cryopreservation. And I will present you the results of the hCOMET group. But before I start, I need to explain you what is actually the alkaline Comet assay, or this alkaline version of the single cell gel electrophoresis. As you can see on this slide, it is a widely used technique thanks to its simplicity, sensitivity, and expensiveness for gaining reliable results in the assessment of strand breaks and alkali labeled sites, DNA protein cross links, and incomplete excision repair sites. It is a highly sensitive method to detect low levels of primary DNA damage in single proliferating and non-proliferating cells. So you need to have a single cell suspension for this assay. It is a low cost, easily done method, and you can get results in a really short period. It was introduced in 1988, and together with the micronucleus assay, it was proposed as a potential tool for assessment of genotoxic potential of chemicals and carcinogens, and it gained its OECD guideline in vivo in 2014. Basically, the technique, in techniques, these single cell suspension cells are sandwiched in agaroses gel layer on the microscope slide. Usually you have 2 or 3 layers. In the middle or the upper layer are the cells. After that, there is a treatment with lysis solution, which serves to remove cell membranes, cytoplasm, and nucleoplasm, including nucleosomes. Afterwards, there is alkaline incubation for unwinding of the supercoiled DNA, so that the damaged DNA can be, parts of the damaged DNA can be free. And during the electrophoresis that comes after the alkaline incubation, those small parts can travel through the electrophoresis, while the undamaged DNA or the big DNA cannot travel through the pores of the agarose gel. Of course, you can see on this small table some endogenous sources and exogenous sources that can make DNA damage and can be detected with the Comet assay. And with this assay can be even more specific if you add one step with digestion with specific endonuclease, and then you can measure really specific types of DNA damage, mostly oxidative one. Of course, this assay can be used as a DNA repair assay and for challenging assay, but you will hear more about that later. Of course, in each lab, there are different scoring techniques, the use of different gel percentage, timing for electrophoresis, the use of visual scoring or different softwares, or the use of different parameters such as tail length, tail moment, or percentage of DNA in a tail. Is making a little bit confusion between the labs, and so there is a big need to standardize this procedure so we can also repeat similar experiments in all the laboratories, and also we can so that we can also compare the results. That's why this hCOMET group has been developed. As you can see, the chair of the person was Andrew Collins. We had 64 partners from 24 European countries, even 3 Coast international partner countries, and also researchers from Mexico, Cuba, and Brazil were included. We were divided in 7 working groups. And for more information about everything, you can see on our webpage, www.hcomet.eu. We have published really a lot of videos, protocols, guidelines that you can check on our page. They are free. And we have published more than 100 scientific papers, reviews, and guidelines. This was one of our first threefold, so that's why I'm representing you, but I will not talk about the details here. You can see the map of the countries or the scheme of the working groups. So one of the first steps in our group was to create a database, and we have collected the data from more than 9,000 individuals. We have also published protocols, guidelines, editorial recommendations. We have even 3 Nature protocols and manuscripts in scientific reports. This is one of them, Mirta. So if you are interested, you can check it. This is another one for in vitro DNA repair assay. We have compared the results and the use of different biological source material, because in human biomonitoring, you are usually using blood or isolated cell fractions like leukocytes or fresh or frozen blood. We have even published 2 special issues in mutation research. This is one, and this is the second one. We have also made a lot of training schools, short-term scientific missions, videos, and you can check everything again on our webpage if you're interested. So this network was established from the Comet at the beginning, has involved in hCOMET, this co-section, and because it has been finished, all the scientists from the hCOMET are involved in ICAO. ICAO is actually newly formed International Comet Assay Working Group, founded as a specialist interest group of European Environmental Mutagenesis Genomic Society in 2020. And you can see on our webpage the details about it. And usually every second year we are organizing the congress that we like to call workshops, because usually there are some kind of training schools. And we have rescheduled this workshop for the May 2022 in Maastricht, Netherlands, and you are all welcome to sign in and also to sign for the training schools that will be also held there. So let's talk again about my topic of my talk. So in human biomonitoring, you are usually using human blood, and it is usually freshly used. It can be used as a whole blood, isolated leukocytes, peripheral blood mononuclear cells, or cell fractions. But usually, sometimes you need only small samples, or you can use it fresh. Or if you have a lot of samples, it is a timely consuming preparation of isolated leukocytes, PBMC cell fractions. So sometimes it could be really better to use frozen samples with cryopreservation or without cryopreservation. You can see here that you can use whole blood or isolated fractions, but there is possibility when you are using cryopreservation that you will have DNA damage when you compare the results with the freshly used samples. And if you are using no cryopreservative, we don't know for how long can those samples be stored without the DNA damage. And there are not many articles about that. So in our hCOMET group, we wanted also to analyze and also to compare those samples. Because frozen samples, you can choose the time of the day when you will use them. You can use repeated samples. And also you can use those samples from the biobank for reanalysis. And the use of frozen samples is actually opening new fields to use alkaline Comet assay. So with different cryopreservation methods, there is also a problem. So we wanted to compare also different methods, because some labs are storing blood on -80, liquid nitrogen on -20, or they are using different cryopreservative concentrations of dimethyl sulfoxide, that is DMSO, and serum and everything, and also the different concentrations of the cells. So I will just show you briefly few articles from our group that have been published. From 2017, our scientists have concluded that whole frozen blood and PBMC frozen can be used for DNA damage detection with alkaline comet, but in this article they did not check the timing of the freezing. In another article, they have again checked not only alkaline comet assay, but FPG alkaline comet assay and challenging assay with peroxide, with PBMC lymphocytes and buffy coats, with 1, 4, and 12 weeks frozen samples. And they have concluded that at the end, frozen whole blood samples stored in cryostroves can be used, and that the whole blood is the most suitable to be used to perform biomonitoring in humans. I don't have enough time, so I will just show you some pictures that you see that it was really an intensive work, and you can check the articles by yourself. So at the end, again, another group has also used the cryoprotection for 12 months, and they have concluded that it can be used. And to conclude this part with cryopreservative, in 2021, our scientists from our group have published in scientific reports results of the impact of 12-month cryopreservation on endogenous DNA damage. What they have found or discovered when they have compared PBMC and white blood cells is that there was increased DNA damage, and that it was continuous. In PBMC, it started from 6 months up to 12 months, and in whole blood from 4 months up to 12 months, comparing with the baseline levels. They have shown that whole blood cells have that actually PBMC have a slightly higher DNA damage. And in oxidative FPG comet assay, they again demonstrated that PBMC has significant increase in damage cryopreserved from the 8th month of cryopreservation, and that whole blood has significantly higher DNA damage from 4th month of cryopreservation. Now, again, when they have compared the results up to 8 months, the results were similar for both whole blood and PBMC, but after that, whole blood had slightly higher values. And using calibration curves for PBMC, they have concluded that approximately 2,600 extra lesions has been made at the end of the 12th month of cryopreservation. So another group has checked could PBMC and whole blood, although it is written cryopreserved, they did not use cryopreservation, be used for alkaline comet assay, and can results be comparable with the fresh one. But they have concluded that we should use smaller amount milliliters of blood for freezing. Another group from 2011 showed that for 1 month, if you are freezing it only in -80, not on lower temperature or high temperature, that there is no difference in DNA damage, that you should use only 0.6% of LMP agaroses, not different, because they have used really different percentages. And that you should use a maximum of 250 microliter volumes of whole blood that was frozen, because higher levels of that volume are adding this DNA damaging effect. They have checked that only 5 microliters of your defrozen sample is enough to get reliable results, because there will be more than 5,000 cells with reliable results. Okay, this is just the storage temperature, because they have also checked the oxidative damage. In another article of them, they have checked it for 4 months with 3 washing steps, and gain again similar results that can be comparable with the fresh ones. But after 11 months with washing steps, they have checked only oxidative damage. So another group, that is a group from my laboratory, have checked what is the damage after 1 year of freezing without the cryopreservation. And but we have used for whole blood for whole blood storage EDTA. That is most important, not to use the other anticoagulants, because they are adding the DNA damage values higher. So we have demonstrated that even after 1 month of freezing at -80, the percentage of DNA damage is as in background levels of the control. And also from my lab, the other group has demonstrated that their 5 years of cryopreservative, again with EDTA, on -80 has reliable results and comparable one with fresh samples. So at the end, we have demonstrated that repeated sampling can be done from the same individuals, and that alkaline Comet assay can be used in intervention trials. In biobanks, that you can use it for reanalyzation, that you can maybe use it for individuals to demonstrate whether there is susceptibility, higher one for the cancer risk, for disease development and mortality studies. So I would like to thank you for your attention. And now I need to represent all the speakers that will come after me. So the first speaker after me will be Professor Amaya Azqueta Oscoz. I hope that I have said it correctly. So she concluded her PhD in 2006 in the area of toxicology at the University of Navarra. And for more than 4 years, she was working also as a researcher in the Department of Nutrition of the University of Oslo with Professor Andrew Collins. Nowadays, she has gained the really prestigious Spanish research contract called Ramon Cayal, and she is a professor in the Department of Pharmacology and Toxicology at the University of Navarra. And her area of research is genetic toxicology, and she is a great expert in the Comet assay. She is also great at networking, and she is a part of executive committee of the Spanish Environmental Mutagen and Genomics Society, vice president of the European Environmental Mutagen and Genomics Society, and one of the founders of the International Comet Assay Working Group. And of course, within our cost action, she was the leader of the working package dedicated to protocols and SOPs. She has published more than 120 scientific publications in reputed journals, and some of them are really highly cited. And she has been involved in several national and international research projects. After her, there will be Professor Stefano Bonassi. The title of his presentation will be DNA damage in circulating leukocytes that may predict the risk of death and cancer, results from the hCOMET cohort. Professor Bonassi was born in Italy. He's married with Angela Calvi, and they have 4 children. And he has a degree in biological sciences from the University of Genova, and his specialty is in medical statistics and epidemiology. He is currently full professor of hygiene and preventive medicine at the San Rafael University, and the head of the unit of clinical and molecular epidemiology at the San Raffaele Pisana in Rome, Italy. He has worked also in Genova, previously in Italy, as a director of the unit of epidemiology and biostatistics at the National Research Center Institute in Genova, Italy. And since 2006, he is an adjunct professor of molecular epidemiology at the University of Geneva. His main interest is really huge: the use of biomarkers in human population and clinical studies, particularly the role of chromosomal damage in early stages of carcinogenesis and other chronic diseases. He was a coordinator of many, or was involved in coordination of many international collaborative projects, such as ESCH on chromosomal damage, human and human XL on the micronucleus assay, that is both on blood and buccal cells, on COMET, on DNA damage. He is really active in designing and coordinating epidemiological studies, often based on high throughput techniques, such as gene expression profile and genotype. And in Rome, his research includes research on neurodegenerative disorders, COPD, aging, pharmacoepidemiology, frailty, and rehabilitation. And recently, he started a collaborative project on validation of a system medicine approach for the collection and evaluation of clinical and biological information of patients affected by non-communicable diseases. He is also a teacher of epidemiological methods in several international courses for postgraduate students, and has supervised many theses and postgraduate research projects, not only in Italy, but in Europe. And his research has been funded by several national and international agencies. From 2009 and to 2013, he served as a president of the International Association of Environmental Mutagenesis and Genomic Society, and that is an international association with 6,000 members all over the world. And he is member of the editorial board of several journals in the field of epidemiology and genetics. He is often invited to speak at international meetings, courses, and seminars, and overall has 300 papers and has been cited more than 5,000 times with an H index of 58. And our last speaker is Professor Vanessa Moraes de Andrade from Brazil. I am not sure how to say this, but I will say it. She is from University do Extremo Sul Catarinense. And the title of her presentation will be Brazil Nut Consumption Reduces DNA Damage in Patients with Type 2 Diabetes Mellitus, probably through changes in oxidative status. Professor Vanessa Moraes de Andrade has a degree in biological sciences, master and PhD in genetics and molecular biology, all from Federal University of Rio Grande do Sul, Brazil. And she is currently a professor at the University of Santa Catarina and a member of the board of the Brazilian Association of Mutagenesis and Environmental Genomics as a vice president. She is a secretary of International Working Group of the Comet assay that was founded in 2020. In undergraduate, she teaches genetics and molecular biology in courses such as biomedicine, medicine, and pharmacy. She is involved in research productivity scholarships since 2009 and has experience in genetics with emphasis on mutagenesis, with the topics anti-mutagenesis of medicinal plants and functional foods, occupational risk, environmental biomonitoring, chemical and environmental genotoxicity, toxicity, COMET assay, and micronucleus test. She has published more than 100 articles in international journals. So that is so much from me. Professor Amaya Azqueta, the floor is yours.
¿Ahora? Vale, gracias. No, estaba comentando que como Mirta ya ha dicho, soy profesora de la Universidad de Navarra. Navarra es una de las comunidades autónomas de España. Está al norte, pegadita a Francia. Y aunque se me da muchísimo mejor el español que el inglés, voy a dar esta charla en inglés para que la puedan seguir aquellos asistentes que no sean hispanoparlantes y sobre todo pues mis compañeros de sesión. Bueno, quería aprovechar para agradecer la oportunidad que me ha dado la organización de poder presentar mi trabajo en este foro tan especial. The title of my talk is Technical Recommendations to Perform the Alkaline Standard and Enzyme-Modified Comet Assay in Human Biomonitoring Studies. Oops. Sorry, I don't know, it doesn't work. So Mirta has already talked a bit about the Comet assay, but I'm going just to say some very specific things. The standard Comet assay detects DNA strand breaks and alkali labile site at a single cell level, but the assay has been modified using different lesion-specific enzymes to detect other lesions such as oxidized and alkylate bases, pyrimidine dimers and uracil residues. But the assay also can detect crosslinks and DNA bulky adducts by other modification. This assay can be applied to virtually any biological material from which a cell suspension can be obtained. So it is widely used in genotoxicity testing, both in vitro and in vivo. The in vivo version has an OECD guideline, as Mirta already said. But it is also used in human and ecological biomonitoring, in ecotoxicity, in clinical at research level, and in research, in basic research. So in this talk, I'm going to focus on the standard and the FPG modified Comet assay. FPG is a bacterial enzyme from the DNA repair machinery of the bacteria that is called formamidopyrimidine DNA glycosylase and is used in combination with the Comet assay to detect oxidized purines, mainly 8-oxoguanines. And here we have the protocol. Yeah, I know Mirta has already explained it, but I'm going to go through it once more because it's important to have it in mind in order to follow my talk. So the protocol is quite simple. The first step, in the first step, the cells are embedded in agarose and placed on a microscope slide to form a thin gel. Well, now other supports apart from microscope slides are also used. But anyway, the cells are lysed to form nucleoids, which contain supercoiled DNA. And then they undergo an alkaline incubation and an electrophoresis. Then, after neutralization and washing, the nucleoids are stained with fluorochrome and visualized under the fluorescence microscope. So if the DNA of a cell contains breaks, it relaxes and is able to migrate during the electrophoresis. And it creates images that are very similar to the stellar comets, as you can see in the figure. So the relative intensity of tail reflects the DNA break frequency. The last part of the protocol is the scoring of the results, and this can be done visually or more commonly using an image analysis software. And this software calculates different parameters per comet. And the most used parameters are the percentage DNA in tail and the tail moment. This is calculated per comet. Then the mean or the median of 100 or 150 comets are calculated, and that number represents the sample. This assay was described in the 1980s by Oslin and Johansson in Sweden and by Sin and collaborators in the United States. The protocol of the enzyme-modified Comet assay is very similar. After the lysis, the nucleoids are digested with lesion-specific enzymes. These enzymes will introduce breaks at the site of the specific lesions. And the frequency of a specific lesion is given by the increase in DNA breaks in the presence of the specific enzyme. This approach was described in 1993 by Andrew Collins in the paper you can see in the slide. They were using endonuclease 3, which is also a bacterial enzyme. They were using it to detect oxidized pyrimidines in human lymphocytes and in HeLa cells. Well, I think I'm going to skip this slide because Mirta has already explained the hCOMET project. I just want to say that I was the lead of working group 4, the one dealing with protocols. And the objectives of this working group was the production of guidelines and standard operating procedures for the use of the Comet assay in biomonitoring, and also to identify causes of interlaboratory variability. The COMET assay has a quite high interlaboratory variability, and we have made a lot of efforts to detect which are the causes of this variability and also to decrease it. There are a lot of COMET assay protocols published, and I think we retrieved about 14. And here in this slide, you can see the protocols that has been used in multi-laboratory trials. In the first column, you have the steps of the protocols, and then we have 3 European projects in which the protocol, the COMET assay was applied, the Oscoz, Mutagenesis, and ECBAC. And the last one is the OECD guideline. And if you check the conditions used in each of the steps, you will see that there are a lot of differences among them. For instance, if you look at the agarose, the concentration of agarose used is different among them, the time of the lysis is different, the time of the alkaline treatment is different, the conditions of the electrophoresis also are different, and so on. So in view of these results, we decided to do a survey in the hCOMET to assess the differences in methods. And 33 hCOMET laboratories participate in this survey. And here I'm going to show you some of the results. The range of the percentage of agarose used was from 0.5 to 1.5, being 0.7 the most used. Regarding the duration of the lysis, it went from 1 hour to weeks, being 1 hour the most used. Regarding the duration of the alkaline incubation, it went from 15 to 40 minutes, being 20 the most used. Regarding the electrophoresis, the voltage per centimeter went from 0.7 to 8 volts per centimeter, being 1 the most used. And regarding the duration of the electrophoresis, it went from 20 to 30 minutes, being 20 minutes the most used. So, and this is just some examples. Neutralization and washing was made differently, staining was made differently, even scoring was made very different. So how all these differences in the protocol can affect the COMET assay outcome. So in the next slides, I'm going to give you some examples to answer this question. So let's start with the concentration of agarose. Here in the title, you can see the range of concentration of agarose that was used in the hCOMET laboratories. So here in this graph, we have results obtained after analysis untreated and H2O2 treated lymphocytes using different agarose concentration from 0.4 to 0.95. And as you can see, there are no differences in the untreated cells, but there are a lot of differences in the treated ones. Same results were obtained in gamma irradiated TPH1 cells. In this case, they were using agarose concentrations for 0.4 to 1.3. So final agarose concentration in gels is inversely proportional to percentage DNA in treated cells. And this is something that is expected since low concentration will in a way facilitate the migration of the DNA. Regarding the duration of the lysis step, here we have results obtained after analyzing TK6 cells untreated or exposed to 2 X-ray doses. And they were analyzed using no lysis at all, 1 hour of lysis, 24 hours, and 1 week of lysis. And as you can see, all the results were the same. Surprisingly, no lysis was given the same results as 1 hour, 24 hours, or 1 week of lysis. And this is due to the fact that the alkaline treatment, which is the next step in the COMET assay, can lyse the cells. So in a way, the lysis step is not needed when the standard COMET assay is performed. Of course, it is needed when the enzyme-modified COMET assay is performed because the enzyme needs to access the nucleoids. Here we have also results from untreated and MMS-treated HeLa cells analyzed using different times of lysis, from 0 to 1 week. We obtained the same results from 0, so no lysis, to 1 week, to 1 hour, sorry, but in longer periods of lysis, we can see a clear effect in the treated cells. And same results were also obtained with HeLa cells treated with hydrogen peroxide. So the duration of the lysis has an effect in cells containing a certain type of lesions. A longer period of lysis allows the detection of a considerable great amount of lesions. Lysis is performed at pH 10, and it is known that some lesions are not stable at this pH. So it seems that lesions that are not detected with the Comet assay during the lysis period are converted into lesions that are detected with the Comet assay. So strand-breaks and alkali labilicytes. And the kinetic of that conversion is not known. In the case of the lysis, also the pH has an effect in the results. Okay, here we have, when we are performing the enzyme-modified Comet assay, the enzyme concentration used and the duration of the enzyme incubation is crucial. These are results from TK6 cells and exposed to potassium bromate. This compound induces 8 oxoguanines. And we use 3 different times of incubation, 15 minutes, 30 minutes, and 1 hour. You can see one in each of the graphs, and different concentrations of the enzyme. And as you can see, while the results in non-treated cells, which is the gray line, are not very different, the results in the treated ones, the black line, are very, very different. This type of experiments, well, actually this experiment is a titration, are performed to select the enzyme concentration and the duration, the optimal enzyme concentration and duration of the incubation, in order to detect all the lesions that are present in a cell without having non-specific activity. Here you can see one example of non-specific activity. The concentration is so high that it's going to start making breaks anywhere. So from these experiments, we can choose these 2 situations to perform the Comet assay afterwards. Since we are detecting all the lesions present, you can see a plateau, and we are not detecting any non-specific activity. So enzyme concentration and incubation time are critical, and titration experiments are required to determine the optimal conditions. Regarding the duration of the alkaline treatment, here we have results obtained after analyzing untreated and H2O2 treated lymphocytes and TK6 cells using different times of alkaline incubation from 10 minutes to 60 minutes. And as you can see, well, there is no effect on the untreated cells, or at least the effect is not very clear, but it's a clear effect in the treated ones. And the same results were obtained when using A549 cells. Here in the first graph, the graph A, these are untreated cells. The second one are cells treated with gamma ray, and the third one are cells treated with hydrogen peroxide. And they were using 20, 40, and 60 minutes. And results are similar to the previous ones. So increasing the duration of the alkaline treatment induced an increase in the DNA breaks detected. During the alkaline treatment, all the alkali labilicytes are converted into breaks. And the kinetics of this process is not well known. The temperature also has an effect in the alkaline treatment. Normally, the alkaline treatment is performed at 4 degrees, but some people use 10 degrees or even room temperature. And the temperature used has a great effect also. Well, electrophoresis. Electrophoresis is the key step in the Comet assay. So both the voltage and the time of electrophoresis has a big effect. Here we can see cells treated with hydrogen peroxide. These are lymphocytes that has been analyzed using different voltage gradient from 0.16 to 1.48. And there is a slight effect in the untreated cells, but a clear effect in the treated ones. And the same results were obtained in A549 cells. also treated with hydrogen peroxide. In this case, they use an electric file of from 0.7 to 1.6. So the higher the voltage, the higher is the DNA migration. And this is something that, of course, is expected. Regarding the duration of the electrophoresis, here we have similar experiments, but we change the time of the electrophoresis and keep constant the voltage. And as we can see, there is a slight effect in the untreated cells and a clear effect in the treated ones. And the same was observed in A549 cells, either treated with hydrogen peroxide or with gamma radiation. So increasing the duration of electrophoresis enhanced the DNA migration. And actually, voltage and duration of electrophoresis compensate each other. So within a range of values, of conditions, a strong but short electrophoresis can give the same results as a soft but long electrophoresis. And here the temperature also has an effect, as the same as the alkaline treatment. Normally the electrophoresis is done at 4 degrees, but some people use 10 degrees or even room temperature. Okay, staining and scoring is the last step of the protocol. And it also has an effect in the outcome of the assay. And I'm going to tell you the experience we have in hCOMET. We ran a ring study with 10 laboratories involved on it. The aim of this ring study was to check if potassium bromate was a good positive control for the FPG modified Comet assay. I'm not going to explain about the whole ring study, but within the ring study, reference samples were distributed to the 10 laboratories. These were TPH1 cells treated with a photosensitizer plus light. This is also used to induce atoxoguanine. The 10 laboratories analyzed the samples following a common protocol, and at the end, the coefficient of variation of the results between all the 10 laboratories were 49%, which is quite a lot. Then we do another try, and with the same samples, all the laboratories analyze the samples, but they send the samples to a central laboratory for the staining and the scoring. So the rest of the protocol was done in each of the laboratories, and the staining and the scoring of the comments were done in a central laboratory. And in this case, the coefficient of variation decreased to 28%. This means that the half of the variability was due to staining and scoring of the comments. And this variability can come from different, like the type of dye can affect it, the microscope quality, the microscope adjustment, the aging of the fluorescence microscope light source, in particular when using the traditional mercury lamp. Actually, we experienced in my lab like a couple of years ago that we were obtaining very strange results, and then we realized that the lamp was quite old. And so we made some tests and we realized that we were having a lot of variability because of that. So we have started with a LED lamp, and now everything seems okay. Also, the settings with the image analysis software can affect the results, and of course the method used for scoring. So if we are back to this slide where we can see all the different methods used in hCOMET, even when some of the parameters can compensate others, I think we will obtain very different results. So to finalize, I'm going to give you some recommendations for the good performance of the Comet assay, also to decrease the intra-laboratory variation, and also to be able to compare our results with results from other laboratories or other institutions. So first, keep a constant time of lysis. As we have seen, the lysis, depending on the lesion we have, has a great impact on the results. And the lysis step is a moment in the Comet assay where you can stop. So you can stop and you can start again in an hour or in 2 hours or the next day. And this sometimes when you have a set of experiments, you realize that you have used 1 hour in some of the experiments, overnight in some others, 24 hours in others, 4 hours in others because you went for lunch or wherever. So this will increase a lot the variability, the intra-laboratory variability. So keep a constant time of lysis. So choose one and keep it. Then use a constant volume of the electrophoresis solution. Of course, giving a reasonable depth of solution of the gel. And this is important. We normally take care a lot of the milliamperes, but that's not important anymore. As long as you reach the voltage you want, the milliamperes are not important. So just keep the same volume, a constant volume of electrophoresis in all the experiments within a set of experiments. Apply cells at optimal densities. This is done to avoid overlapping. Normally, the highly damaged overlapping tends to overlap more than the non-damaged, and you can have some bias in the results. If possible, use a voltmeter to measure the voltage gradient across the platform, since it will give you the real value. Consider using mini-gels to maximize the number of samples that can be processed in one experiment. When using the enzyme-modified Comet assay, perform a titration experiments to determine enzyme concentration and incubation time. So even, do it in your lab with your equipment and with your protocol. Even if someone else from another lab gives you an enzyme and explain the conditions to use it, it is better to perform titration experiments in your lab, in your conditions with your equipment. Include reference standards in all the experiments. Reference standards are cells with a known level of damage. And it's easy to prepare reference standard. You just have to treat cells with, methylmetanosulfonase, for instance, or hydrogen peroxide, and then you freeze the cells. And then each time you are going to perform one assay, just take one and include it with all your samples. And then you can check if one experiment went wrong. Code the samples so that scoring can be done blind. It's just to avoid bias. Calibrate the assay against DNA breakage generated by ionizing radiation to allow the calculation of real break frequency. This is very useful for comparing results between laboratories. It's an approach that is not very used, but I think we should really use it if we want to decrease the intra-laboratory variation. And then the last one, follow the Mirta paper for prescribing Comet assay procedures and results. Mirta has already talked about the Mirta paper. It's a consensus statement among, I think, more than 30 researchers from different institutions and countries. And the title of this consensus is the minimum information for reporting on the Comet assay, recommendations for describing Comet assay procedures and results. It's published in Nature Protocol, but it's open access. And it's good when you have to read a paper in which the Comet assay is described. If it is well described, you can even guess how is the sensitivity of the assay that they use. And you can even make comparisons with your data or with other data. So thank you very much for your attention. Muchas gracias por vuestra atención y estaré encantada de contestar a las preguntas en la sesión de discusión. Muchas gracias.
Yes, I'm sharing the screen. Okay. Okay, can you see the screen well? Hope so. Excellent. So now we change a little and we move from laboratory to the, say, to the real life. in a way, from laboratory to epidemiology. In a way, because the hCOMET project has an important part dealing with molecular epidemiology and epidemiology, which is moving from single cell to population. And obviously, we have a much more variability dealing with people, but more numbers in a way. And the beginning of the study, actually, in a way, we had some aims and priority when we started the project. And some of them were just dealing with to increase our knowledge about the assay. And our knowledge of the assay is dealing, of course, with what the critical question, for instance, it was, what's the basal, the background, the DNA damage that we can measure? And can we compare, can we have a reference value? That would be really important, just to compare, to increase the possibility to compare the results among different laboratories, and even to try to link the extent of the damage to the outcome, adverse outcome that we observe. Obviously, there was another item, which was to understanding the role of different technical features of the assay, and I'm sure that Professor Azqueta has presented very well and it's given a large information about the role of different technical steps. And then finally, what I believe that is an important challenge as well was to understand if what we measure in people which are healthy, if the extent of DNA damage that we measure in healthy people can predict long-term events like cancer or mortality. And I won't spend more time on describing the project. Let me show another map of the planet, showing this is the map of laboratories of the countries contributing data to the hCOMET database, which is quite exciting because that you see that it's really a global contribution. And even though the project was funded by European Community, the group was really international, including an important contribution from the Latin America. And I cannot resist to show you a picture of some selected members of the group. So the point when I said that epidemiology can say something important is that because size matters. And size matters because we are able to put together almost 20,000, as anticipated by our chairman. We put together nearly 20,000, the results of a Comet assay measured in 20,000 subjects, which is a very huge number for molecular epidemiology scale. And 13,500 had valid results that we can include in the statistical analysis. Data comes from 133 papers for more than 100 studies. And even when we cut down the whole amount of data to different predictors of use for the Comet assay, we see that, for instance, the tail intensity, which was clearly explained by previous speakers, is the most common, the standard predictor, has more than 8,000 subjects. So we could make statistics out of 8,000 subjects. But also the other descriptors, tail length, moment, and even visual scoring were provided statistics based on a large amount of people. And I wanted just to stress that these results have been at least the first part, the one explaining the variability of the assay and the results from this large database has been recently published by a paper, which is an open access paper, so you can see it. The first author was our chairman, and you see that's more than 100 authors. That means which was really representative of laboratories working in the field. And the paper was published on the mutation research. One of the results was just to show the baseline level of the results in study groups and in controls. And the common feeling now is that you cannot use the Comet assay. One of the big limitations of the Comet assay is the variability. And as you can see, actually, there is actually a real variability. Here we have the variability in tail length. Each box it represents one laboratory. We have the median value and the 25th and 75th border boundaries. And you see that here is much larger variability. Some other laboratories has lower. Tail intensity, there is much more laboratories as a lower variability. Tail moment and visual scoring. Visual scoring was very, very variable. And for this reason, we did not provide major statistics. But we did it for the other, with the idea that at least we cannot just define a precise level, but we can identify an interval in which most laboratories found their baseline DNA damage results. So this is very useful because each laboratory starting new in the field know that their result should fall in that interval. Otherwise, maybe there is a problem, or maybe a very difference in the population which has been investigated. But in general, we see this. And these are the observed data. I just stripped on the model data that come from a model which included random effect. That was a mixed random effect, a log linear model. So thanks to this modeling, we were able to take into consideration confounders and come out with intervals. For instance, for tail intensity, the median value is around 3.9, and the interval in which most part of the result fall down is between 1.8 and 7.7, which is quite a large interval, but it's pretty useful because it provides a reference. And to play with so many data allowed us also to measure the effect of important host factors like age and sex. And that was quite clear that there was a trend by age of DNA damage, or that males has a significantly higher level of DNA damage as measured with the Comet assay. And this is quite important for several reasons, even for designing the study and for understanding what is a confounder. Again, there is another important issue that we wanted to evaluate. I mean, a Comet assay is a good biomarkers for exposure, and that was not easy to put together different specific exposure, but we were able to understand. We wanted to understand if the reason for which the study was designed, the aim for the study was designed, was actually reached. I mean, in generally, most of these studies were designed to understand even exposure to genotoxic or DNA damaging agents actually turned out to have a higher level of Comet assay. And this happened quite often. And you see that there is this is a typical meta-analysis, what is called a forest plot. Here there is the estimate, the point estimate of effect and the confidence interval. You see that most studies are over the level of no effect. and some other studies falling significantly below, in many cases, investigated antioxidant treatment. So that was actually expected. So from this result, it comes out quite clearly that the COMET assay is an excellent marker for exposure once we take into consideration the variability, at least some variability due to host factor, to confounding factor, or to technical factors. But the big challenge, the big question was, but with the DNA damage, can we predict long-term effect? Can we predict the cancer? The people that now they are healthy, but they have very high level of DNA damage, have a higher risk to die or to get the cancer. So that was very important for several reasons: mechanistic, legal, public health, and so on. So we designed a hCOMET cohort. So the idea was to perform a retrospective historical cohort study, and we were able to went back to the laboratories to take the results of the study and to follow up the people that received a COMET assay evaluation in the past. So the idea of the historical cohort study is like to do a prospective study, but it's like you start now that you have the results, that you have cancer, you have mortality, and go back to the old time and see what was the level of DNA damage in people that contracted a cancer or die of cancer. So this is the design that has been used several times in the past, based on the idea that, okay, you start on genetic background and exposure. The combination of these through an intermediate step induce cancer or chronic diseases. The idea is that we can use some surrogate which are associated with the effect, with the exposure and genetic background, but they can also predict the cancer. And this has been done with other biomarkers. And one of the first was the chromosomal aberrations, that there was a number of European studies, and that revealed an increase of cancer in, there was more or less a 40% higher risk of cancer in subjects in the highest tertile of chromosomal aberration versus those in the lowest tertile when they were young, when they were healthy. The same results were replicated in the micronuclei with the human studies, and you see that the study group was a little smaller. The previous one on chromosomal aberration was about 22,000 subjects. Here was based on nearly 7,000. The results were, however, quite similar, showing that subjects with the lowest level of micronuclei when they were young, when they were healthy, is much better than those with a high, low, or high. In this case, unfortunately, results were not perfectly showing a dose response, but it was quite clear that the healthy subject with high level or medium level of micronuclei has a much lower, much worse probability of surviving than people with a good genetic stability. I mean, low level of micronuclei. So we did exactly the same design within the hCOMET group. These are the laboratories that contributed. There was, most of them were from Europe. There was also a large Brazilian laboratory, mainly from Italy, Poland, Russia, Norway or Slovakia, and Spain, Croatia, Serbia, Hungary. Overall, we had put together only 2,150 subjects for a total of 293 subjects dying of any cause. Unfortunately, the number of cancer cases was not so large, so results for cancer were not very stable. the total of person years was about 22,000 person years for a 10 years median follow-up. The results, however, are quite intriguing. And you see that subjects, this is the higher, the survival curves show clearly that subjects with the lowest level of DNA damage when they were healthy has a much higher probability to survive, whereas people with the highest level of DNA damage are those with the highest mortality, the lowest probability to survive. And these results are quite significant, highly significant. And you see that results are not that different from chromosomal aberrations. And subjects in the highest tertile of DNA damage measured with the Comet assay has about 38% higher probability to die of any causes. We try to remove people dying of accident, and this result, this hazard ratio increased, but the number got smaller. So we prefer to present the overall picture. And you see that the result is significant. So the results are, I'm just approaching the end to say results about the link between DNA damage measured with the Comet assay and the risk of mortality is quite strong. Obviously, the design and the study has several weaknesses, so it's not a definitive result, but it's quite an important result. For what? The idea is that we want to, at least for epidemiologists that love dealing with mortality, with diseases, what we really want to do is to predict diseases. Picture. And therefore, we published together in this month with Michael Fennett from Adelaide, a paper, a position paper, just to try to validate what could be the process for a biomarker from the laboratory to clinical practice, to individual risk assessment. And actually, it's quite a long way. And we make the example of cholesterol that from the laboratory is now currently used in clinics. And one of the important critical steps to validate these biomarkers, including Comet assay, is prospective studies, cohort studies, and especially randomized controlled trials. And if I have to say where we are now with the Comet assay, and I think that we are here, we are trying to make a transition from group level to individual level. Now we need really to validate further with other cohort study the link in between DNA damage in healthy people and mortality and survival and cancer, and be ready for starting with a controlled trial. I mean, to see if actually controlled trial is the proof, the real proof that things works. If we can, for instance, reverse DNA damage, do we improve survival? This is the next challenge. Next challenges, which are also what are the results that the application to real life of the Comet assay results. One is a personalized prevention, of course. We can start from bench to factory, to real life, actually. Can we personalize prevention and to apply specific preventive intervention to specific people depending upon DNA damage? This is a big challenge. Another is individual risk assessment, of course. And even more practical, what are the legal issues dealing with that? I mean, if we can demonstrate that DNA damage in healthy people, in exposed people are higher, so employers has to I mean, to do something. For instance, if our people working in a factory and they are exposed to some carcinogens, and we found them with a higher Comet assay, with a higher DNA damage, should we do something immediately? Or in 10 years, when the people will develop cancer, there will be a legal issue because you knew in advance. So, but I mean, this is quite a challenge. And there are other challenges. For instance, people has been exposed in the past to carcinogens, or former smoker, or former alcohol drinker. Can we monitor them using a risk factor as a Comet assay? Wow, this is a number of big challenges. And I think that we have plenty of years in the future to try to address that. Thank you very much to the chairman for the presentation and the introduction, and thank you for listening. I'll be happy to take questions at the end. Thank you very much.
Well, thank you all for these really nice talks. So we are now open for discussion. We are in time. And until we get those questions, I have a question, Vanessa, for you. I mean, do you want a little bit of rest?
I could ask.
I can ask. Okay, then. Well, why did you choose 6 months? Do you think that you would see, for example, DNA damage results in micronucleus and alkaline Comet before those 6 months, or?
Actually, before this study, we performed another study with vitamin D supplementation, and was performed during 2 months. And the results were good, but we start to thought that maybe during more time of intervention, the results could be better. So, and especially in this study, we are not talking about a compound like vitamin D, but we use a food. So we thought that should be better to use for during more time the intervention, as we are talking about consumption of food and not just to the selenium compound isolated. I think this was the motive at that time.
I have another question in relation to that. Which other compounds can you find in that mat?
And other compounds?
Other compounds apart from the selenium. Which other compounds you can find?
Some phenolic compounds, fibers, vitamins, also some amino acids, and others minerals such as zinc and potassium. But for sure that the most important and the most papers that talk about Brazilian nut is related to the very large amount of selenium.
Okay, thank you.
I would just like to make a comment on Vanessa's presentation, because I mean, it is really interesting. Those are really interesting results, because also with the micronucleus assay that was not a part of this like hCOMET symposium, you have got the lowering decrease of the results, and you have also shown that those people have higher DNA damage levels than those like normal values that even Stefano was included to estimate what are the normal levels of the damage. So really nice.
Thank you.
So we have questions. So I will, if you cannot see them, I will read the first question. So everybody who wants to answer can be can include. So the first question: because of all the variables to control for during Comet assay, when I teach about it in my genetic toxicology course, students question its use quite a bit. What is your experience in this regard? Amaya, do you want to?
Yeah, well, I don't see the Comet assay as a very difficult technique in comparison with other assays. Like, for instance, I'm thinking now, we are now using the most lymphoma assay. Oof, I mean, so for me, it's, yeah, you have to control some variables, but it's not like a difficult thing to do. It's just you have to take care of the pH, of temperatures, and of times, and the rest is like change the slides from one solution to another. So for me, it's not. I wouldn't say that it's a very complicated assay. So for me, the use in genetic toxicology, I mean, it's obvious. I mean, not in regulatory. I mean, in regulatory is only the in vivo COMET assay that is nowadays used. The in vitro has a small space because in the EFSA, I think, recommend the in vitro COMET assay in combination with enzymes to check the mechanism of action of nanomaterials, for instance. That's the only small space of the in vitro COMET assay in regulatory. But I think if you look in the literature, a lot of people use the COMET assay in genetic toxicology. And as I said, I don't think that it's a difficult assay. It's an assay that you have to understand. So if you understand what you are doing exactly, if you understand what will happen if I increase the time of electrophoresis or if I decrease the time of lysis and so on, I don't think it's a difficult assay. For me, the most difficult part is the scoring. Because, I mean, it takes a long time to score. If you don't have an automatic image analysis system that are quite expensive, it takes a long time to score the results. So for me, that's the bottleneck. The rest of the assay is you just have to understand it. And to be careful, but you have to be careful in the rest of the assays also. So that's my experience. So.
I would just like to add that also our Mirta paper that we have published about the protocol can help also to minimize those variables so that the labs can also repeat the research and have the similar results. So that was the reason. So the chemicals that you are using from the manufacturer, the percentage of agarosis and everything, that's what we are dealing about in our hCOMET project. But if you have one standardized procedure with the percentage of agarosis that you are using, the timing and everything, I mean, it's not so difficult, and I mean, the results are reproducible.
Yeah. One thing, let me just add one thing, because he's using, that person is asking about the use in genetic toxicology. Well, I think the COMET assay is a very flexible, I don't know how to say the word, you can play with it and you can make modifications to detect other lesions. For instance, with the use of enzymes, you can detect like oxidized, alkylated lesions, uracil residues, and so on. There are other modifications to detect crosslinks that are not easily detected with other techniques, and also to detect bulky adducts. Well, the version to detect bulky adducts needs to be validated, but it may work. And if it works, it will be very important, because nowadays, like the detection of bulky adducts is very complicated and it's done. It's a place, but we need to work a bit more in the standardization and in the validation.
Okay, thank you for the answers. Okay, first one comment. So Vanessa, to you, that selenium, as many other micronutrients, is very important in diet, mainly as an oxidant. And congratulations for your research. So that was a comment.
Thank you.
And now we have a question for everybody can talk. So do you think, Stefan, are you listening? Do you think that, as it seems to be the case with the cancer, other diseases like rheumatoid arthritis could be diagnosed early with the use of hCOMET?
Yes. Sorry, I was just sharing one moment more my screen, because actually the idea of linking. Obviously, there are different interests, and the COMET assay and the measurement of DNA damage is really a big issue. So there is interest from people working the mechanisms, and even for in vitro studies. There is people working, for instance, in the prevention of diseases and on the early detection of exposure. And obviously there is also who is more interested in clinics, maybe in the use of this kind of information for the early diagnosis, or maybe even for a prognosis, or for the identification of people that can be treated with a particular treatment. And the COMET assay is moving in this direction. So now we have collected a lot of data. We are trying to, we have created a network, and with this network we are assembling information about the link between DNA damage and disease. And this is extremely important in this moment to see what are the diseases which are most closely related to DNA damage. Is I would say quite a lot, including the rheumatoid arthritis, because of course they are on inflammatory basis, and mostly diseases on the inflammatory basis have the basis and oxidative stress in most cases tends to be traced back to DNA damage. And a similar pathway has been done with other biomarkers. For instance, with the micronuclei, we have just published a special issue linking the micronuclei to diseases, to human diseases. For instance, you see here that in that case, we have seen that there are some cases where micronuclei can be used, like for monitoring of genotoxic exposure, or deficiencies of vitamins, or spleen function, or radiation biodosimetry. Then there is a number of situations in which that's what evidence is quite inadequate, but we are getting closer. We are accumulating evidence, like the possibility to predict for an early detection or a susceptibility of cancer, cardiovascular diseases, accelerated aging. For instance, we have a number of studies on frailty. And most of these situations most likely can be reproduced with the COMET assay. And we are a little bit a step backward with respect to the micronuclei on this regard. But I believe that what applies for the micronuclei and other biomarkers of the DNA damage can theoretically apply for COMET assay. And we are trying just from this day, trying to put together evidence and just to move in that direction, because that would be really great, at least from my point of view, to use this kind of information of a very basic damage for clinical purposes. There are several clinical diseases, just thinking about neurodegenerative diseases or even psychiatric diseases or whatever, in which there are frailty is one of the reasons, and in which we have just clinical criteria for the diagnosis. What if we can add also molecular criteria? That would be our dream.
Okay, thank you. Somebody else wants to say something? I don't think that we have questions more here. I don't see them. So do we have some kind of message for takeaway? We have a few minutes more before we finish. Okay, so no more questions.
I have my take-home message.
Yes.
Is about, okay, is about the power of pooling, the power of international collaborations. And maybe international, but maybe national or maybe continental. I'm thinking about, because now our audience in the Latin America, when there is so many groups doing wonderful research. I've been visiting when I was president of the MGS, sorry, and I met many communities in Latin America. And in many cases, you have small studies, small resources. And the idea, of course, I mean, to design new studies is really powerful because you have new evidence. But there is so much that has been collected so far. And the idea of pulling together data for several purposes, just for validation, one assay. I mean, the idea of pulling together data for validation purposes is great. Or just for discovering, there are several exposures that we don't know yet, because we have just, when Mirta thought about nanoparticles, that's a good example. We have small groups, but if you have the possibility to pull together people exposed to the same conditions in different countries, that would be better, because we have numbers that allows us to say something. Because otherwise, very small studies has a little power to say something new. So, and collaboration, creating database, creating collaborative studies provides big databases from which you can say a lot. I mean, from the hCOMET database, we probably say much more than that. And if people is interested, can access this data because they are free. This is another important issue. And finally, it creates an increase in culture. I mean, people exchange ideas, exchange knowledge, and this is a great, great opportunity. So if you have the opportunity to participate to collaborative studies and to pool the analysis, do it, because there really is a unique opportunity.
Yes, of course, that was something that I wanted also to say. And also, please visit our page, ICAO page, and the WWH-COMET EU page, because besides those videos, there are also contacts from each person that was involved in the project. If you want to contact somebody, make collaboration, make questions, visit the lab, or that person comes to your lab to teach you some things. I mean, we are all open for this, because together we are stronger, as you see, creating big databases and connecting all over the world. So in my name, I would like to thank you all. I am letting Vanessa and Amaya to say the final words.
Yes, just a final word that I remember now, because I explained that the ICAO-G, that is the International Comet Assay Workshop Group, was created after this cost action, and it was the start was in relation to the European community, but now the Latin American, the Brazilian, all the people that are associated to a scientific association related to toxicology. For example, in Brazil, we have the Mutagenesis Brazil. In Latin America, we have ALANCTA, that is the Latin American Association of Mutagenesis. And if you are participating actively in the even association, it doesn't matter, European, and you are also automatically associated to the IMGS, as Stefano say, that is the International Association of All Societies of Mutagenesis. And so automatically you can ask to join us as a full member. So we invited you to go to the webpage and see all these explanations, and also send an email that me or Sonia, we will answer and explain to help how to join us, not just a viewer page, but also a full member to participate in this in all these projects that Stefano told. Thank you, and sorry to.
Thank you, Amaya.